Hey everybody, today we are going to start with the chromosomal theory of inheritance. So now this theory or hypothesis, I guess we are calling it a theory still, early 1900s, two scientists, Sutton and Boveri, were using some advanced cytological techniques at the time, so working with microscopes, and they were following or studying meiotic cells, and what they noticed was that these structures called the chromosomes occurred in diploid organisms, occurred in pairs, and they segregated during meiosis. So they were acting a lot like Mendel's unit factors, which we know as alleles, and Mendel proposed that they occurred in pairs in the diploid pea plant, and they segregated from one another during meiosis. So Sutton and Boveri came up with the chromosomal theory of inheritance, which essentially says or proposes that Mendel's unit factors are the chromosomes, or chromosomes are Mendel's unit factors. And scientists, of course, were skeptical at first, but later on, Thomas Morgan, as we saw last week, showed that traits correlate with movement of the X chromosome. Now, this was the white eye mutation, moved with the X chromosome. And after that, he showed that genes using the white mutation and mini wing and the yellow body mutation showed that genes exist in a linear order on chromosomes. Okay, so after the chromosomal theory of inheritance, after Thomas Morgan's experiments, scientists were pretty convinced that genetic information is found in chromosomes. So now the big question was, what are chromosomes made of? Okay, so scientists quickly determined that chromosomes are made of mostly proteins, or protein and DNA. So okay, if chromosomes hold the genetic information, which is more important for that? Is it protein or is it DNA? Now, this was early 1900s, and at that time there was a lot of interesting research being done on proteins, which were made up of 20 amino acids, and not so many exciting discoveries with respect to DNA, which was only made up of 4 nucleotides. And we'll talk about the structure of nucleotide later on in one of the next videos. So it was thought that, well, the genetic information, whatever encodes genetic information, is probably going to be complicated. And so 20 amino acids is more complex than 4 nucleotides. So maybe proteins, or the protein component of chromosomes, is responsible for holding the genetic information. Another thing working against DNA at the time, or at least working against DNA in the minds of most scientists at the time, was this tetranucleotide hypothesis. Tetranucleotide hypothesis. Now, this hypothesis was, at the time, it was widely believed that the levels of adenine equal the levels of guanine, which equal the levels of cytosine, which equal the levels of thymine in cells. And this led to this hypothesis that these nucleotides formed this tetranucleotide structure that essentially served as a scaffold. If you could stack many of these on top of each other, this could serve as a scaffold for various proteins that held the genetic information. Now, with these being equal in number, it was thought that, okay, this is very simple, a repeating structure. Oh yeah, well this could perhaps be a scaffold for holding the proteins, which probably hold the genetic information. At least that was the thought at the time, or at least that is my understanding that was the thought at the time. So scientists were focused on trying to figure out how traits are passed from parents to offspring through the chromosomes, and with respect to the chromosomes, how the proteins are arranged in chromosomes. And if we could figure that out, well, then you would know how genetic information is passed from parents to offspring. We now know that that is not true, right? DNA is what encodes genetic information. For most organisms, there are some viruses that use RNA, that have RNA genomes, but we'll talk mostly about DNA, organisms that use DNA to encode genetic information. And so the tide turned against protein and towards DNA with some experiments that were performed, or at least reported, in 1927 by Fred Griffith. Now, what he did, he was working with Streptococcus bacterium, and he was working with two strains, so 2R and 3S. Now 2R is what we're going to call non-pathogenic, non-infectious in mice. So it's the non-infectious strain. And if you were to plate this out on a petri dish, it would form these little rough pinpoint colonies. And I'm told that the edges are rough looking and they are small. We'll call them pinpoint. And let's say that's where we get this R from. I don't know if this is true. Maybe this is a legend, but the R stands for the rough edges of these tiny colonies. Now, if we were to plate 3S out on a plate, again, this is just a different strain of Streptococcus, we would get these smooth spreading colonies, smooth edges. In this one right here, let's say smooth and spreading. Now, okay, this one non-infectious, and this one is infectious in mice. Okay, so Griffith performed a number of experiments, and some of them, I should say, worked like this. So he took 2R, injected into mice, and when he did that, the mice lived, right? Because this is not the infectious bacterium. He took 3S. He injected mice with 3S. And here, the mice died, which is what we would expect, right? Put 3S in mice, the mice die because 3S is the infectious one. Now he heat killed the bacteria, injected the heat killed 3S into mice, and the mice lived, which indicated that, okay, the 3S, the bacteria needed to be alive in order to cause the infection in the mouse, in a mouse. So then what he did was he mixed heat killed 3S with live 2R, injected the mixture into mice, and the mice died. each mouse that was injected died. So, and when he took the carcasses and he isolated Streptococcus from the bodies, what he found, well, he found the 2R, right? He injected 2R into there, and he also found living 3S. Now, Griffith was I think it was a biochemist, and he was mostly interested in this phenomenon from a biochemical perspective. He came up with this idea that there was some compound called the transforming principle, and what it did was it transformed 2R into 3S. And this compound was found in the I guess, dead cells, the dead 3S cells. And there was some compound there that was switching the 2Rs into 3S. And my understanding is that he didn't really look at it from an inheritance type of perspective. He kind of looked at it like there was some switch that was turned on in the 2R cells that turned them into 3S. And but we now know that, no, it was a genetic change in the bacterium. And as we talked about, I think in lecture 9, and this might be a coincidence, but it could be where this word comes from for transformation, because it accurately describes what's going on. The bacteria are picking up something, importing something from the environment, in this case naked DNA, and it's causing a transformation of 2R to 3S. And that transformation is heritable, such that the daughter cells that derive from the transformed 2R to 3S, the daughter cells that derive from the 3S cell are also 3S cells. Now, I'm getting ahead of myself. So, after Griffith's experiments, scientists were focused on what is the transforming principle. I told you it was DNA. You probably knew it was going to be DNA, but they didn't know. They didn't know what the transforming principle was. They didn't know what component in the dead 3S cells was transforming the 2R into 3S. Excuse me. So scientists set out on a search to try and figure out, what was the transforming principle. And there were some key advances that were made in this sort of line of experimentation some advances that were made that allowed scientists to finally discover what the transforming principle was. Some of those advances were scientists figured out that you could heat kill the 3S bacteria, and then what you could do is mix the heat-killed 3S cells with 2R bacteria in a test tube, and then plate those out, and then you'd get the transformation occurring. So you didn't need the mouse body for this to happen. Let's get another pencil here. Sorry about that. Here we go. So you didn't need a mouse body. You could do this. This transformation could occur in a test tube. They also found that if you took the heat-killed 3S and you disrupted the cells, so maybe you don't even need to heat kill it. You can just disrupt all the cells, but maybe you better kill it with heat to make sure no living cells get through this disruption process. But you disrupt the cells to create a lysate. So we'll call this the 3S, heat-killed 3S crude lysate. So just the broken open cells, I guess whatever is inside the cell, just kind of spill that out and make a suspension of all that stuff. And you can mix that with 2R in a test tube, and you can get the transformation process occurring. So no living 3S's went into the experiment, but both 2R, living 2R and living 3S colonies came out. So with this experiment, we know that the transforming principle is in here. So essentially, it's just looking at all the components inside the heat-killed 3S crude lysate and finding out which one is responsible for transformation. And then you'll have your key, your molecule that, you know, carries genetic information. Is it protein? Is it DNA? Is it RNA? Is it cell walls? You know, scientists, they didn't know back then. So 10-year experiment, 10-year process or line of path of research was performed by Avery, MacLeod, and McCarty. Now, I'm terrible with these McCarty names. I don't know if this is, if I have an extra C here or not, but again, I never ask you guys to remember these names. I'm putting them in here just to help tell the story here. But okay, so these scientists worked for 10 years to figure out was the transforming principle protein, was it RNA, was it DNA, and how did they do that? So they took the crude lysate, 3SCl, for 3S crude lysate, and they purified it. So they did an initial purification. So I'm going to call this 3S pure lysate. And what this means was, let's say they remove insoluble material. So things that don't dissolve in a water-based solution, like cell walls, for instance. So they got rid of all the insoluble material. And let's call this a pure lysate. It's like a semi-pure lysate. Now, they took that and they treated that with proteases. So proteases are enzymes that degrade proteins. So they found a lot of proteases. I'm not even sure. They may have even had to isolate the proteases themselves and had to purify them and make sure that they have pure proteases. So this line of experimentation took a long time. And it may have been because they needed to discover this. I'm not sure about that. In the future, one of the reasons why I'm making these videos is so in future versions of this course, students will watch the videos before coming to class, and then we'll look at some of the original papers done by scientists like Avery, MacLeod, and McCarty. But anyways, they found the proteases, and they treated the pure lysate with proteases, and that produces a 3S pure lysate minus proteins, right? They degraded all the proteins with proteases. They took that and they treated it with RNases. RNases. It's common to write it like this: capital R, capital N, capital A, and then SES. Now these are enzymes that degrade RNA. So we have 3S purified lysate minus protein minus RNA. Then they took that, and you can probably guess what they did next. They treated that with DNases. So that gives them 3S purified lysate minus protein minus RNA minus DNA. So now they have all these different samples, right? They have the pure lysate, they have the pure lysate without protein, pure lysate without protein and without RNA, and the pure lysate without protein, without RNA, without DNA. I don't know what's left in here. Maybe some sugars or something. And then what they did was, when they took these things and they mixed with live 2R to try and perform the transformation reaction, you know, in this case, let's see, they got transformation. Okay, live 2R and live 3S came out. This one right here without protein still worked. So this tells us, well, proteins aren't needed for the transformation reaction, so the transforming principle is not protein. This one right here, no protein and no RNA, still worked. Transformation is still occurring. So whatever is causing 2R to transform into 3S, it's not protein, it's not RNA. This one right here, you can probably guess, transformation did not work. So once you remove the DNA, transformation does not work. Only 2R cells were found. Okay, so DNA is the transforming principle that Fred Griffith first didn't identify, but he found the transforming principle, and it contained the DNA, and the DNA was the thing that was transforming 2R into 3S. And Avery, MacLeod, and McCarty examined the crude lysate from the 3S cells and determined that of all the components in the crude lysate, it was the DNA that was doing the transformation. So this was the strongest evidence to this point in time that DNA was the molecule of inheritance. So DNA stores the genetic information. It stores genetic information. And then there were some other experiments that were done which supported that hypothesis as well. So we now know that DNA is the genetic material of most organisms. Like I said before, some viruses have an RNA genome, but most organisms use DNA. And in the next video, we are going to, well, you know what? Not yet. Let me tell you a little bit more about DNA. So now that scientists knew that DNA was the genetic material, they wanted to figure out its structure. So if you could figure out its structure, then maybe you could figure out how, DNA stored genetic information or encoded genetic information. So one of the key discoveries was made by Edward, I think it was Edwin Chargaff. His studies allowed us to come up with Chargaff's rules, and he essentially refuted the tetranucleotide hypothesis, and he found that levels of A were, equals the levels of T inside cells, and the levels of adenine equal levels of thymine, and levels of guanine equal levels of cytosine. So this is very different from the tetranucleotide hypothesis, which said A equals T equals G equals C. Okay, so levels of A equal T and levels of G equal C. Then another key major discovery was made by Rosalind Franklin. So Rosalind Franklin was one of the best X-ray crystallographers of her time. And she was studying DNA, trying to figure out the structure by performing X-ray crystallography. And I wish I knew how this process worked, how to interpret these graphs or radiograms or whatever they're called, how she interpreted them, or how anyone could interpret them. But I have zero knowledge with respect to this. But I know she came up with this. She was one of the best of her time, and she was working using X-ray crystallography to figure out the structure of DNA. And her boss was a scientist named Maurice Wilkins. And I'm not sure of the relationship exactly, but I guess Maurice Wilkins was the head of a big lab where a lot of very capable scientists worked. So he was the supervisor in charge of a lot of other scientists who were very capable, one of them being Rosalind Franklin. Now, Rosalind had some amazing data. This is very common when you're working in research. You're spending all your time doing experiments. You hardly have enough time to interpret the data you're generating. So the story goes that Maurice here shared Rosalind's X-ray crystallography data on the structure of DNA, some of her hot off the presses data, without Rosalind's approval. Shared them with Watson and Crick. Now, Watson and Crick were working on the structure of DNA for, I think, for a couple of years, and they were taking mostly a theoretical approach, measuring bond angles and structures of nucleotides, trying to fit these things together, trying to predict how DNA could possibly encode genetic information. And I think there was some crystallography data available, and they're trying to interpret those things, and they can't find something that makes sense. The story goes, and I think this was in Watson, I think, wrote a book called The Double Helix, and he recounts it as, when Maurice showed Rosalind's data to these two, they knew exactly what they saw that, and that was the missing piece. They saw that, and they were able to quickly put together, interpret the data, and quickly make sense of some things that they were thinking about for a long time. They saw that, and they said, "Oh, okay, now that's the missing piece. That's the information we need to make our model work." So with that data, they were able to come up with the Watson and Crick model of DNA. So, and as I said in lecture, possibly if there are such things as alternate universes, there may be an alternate universe where Maurice Wilkins does not share this data with Watson and Crick, and then Rosalind Franklin gets most of the credit for coming up with the structure of DNA. And instead of the Watson and Crick model of DNA structure, we have the Rosalind Franklin model of DNA structure. But in our universe here, it didn't work that way. The Watson and Crick got most of the credit. Rosalind Franklin and Maurice Wilkins were co-authors on the paper of this study. I believe they were co-authors on the paper of this study. Unfortunately, Rosalind passed away from ovarian cancer not long after the structure of the DNA was published. And so when the Nobel Prize was awarded, Watson, Crick, and Wilkins were awarded with the Nobel Prize for the discovery of the structure of DNA. The Nobel Prize cannot be awarded posthumously, so Rosalind wasn't considered for the Nobel Prize. Okay, so in the next video, we're going to take a look at the Watson and Crick DNA model.